Breathing and exchange of gases. Oxygen is utilized by the organisms to indirectly break down simple molecules like glucose, amino acids, fatty acids, etc., to derive energy to perform various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reactions. It is therefore evident that oxygen has to be continuously provided to the cells, and carbon dioxide produced by the cells have to be released out. This process of exchange of oxygen from atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Respiratory organs. Mechanisms of breathing vary among different groups of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coeluroids, flatworms, etc. exchange oxygen with CO2 by simple diffusion over the entire body surface. Earthworms use their moist cuticle, and insects have a network of tubes, tracheal tubes, to transport atmospheric air within the body. Special vascularized structures called gills, bronchial respiration, are used by most of the aquatic arthropods and mollusks, whereas vascularized bags called lungs, that is pulmonary respiration, are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds, and mammals respire through lungs. Amphibians like frogs can respire through their moist skin, cutaneous respiration. Sponges, coeluroids, flatworms use simple diffusion, earthworm, cuticle. Insects, tracheal tube, mollusk and aquatic arthropods, and fishes use gills, that is bronchial respiration, and pulmonary respiration by vertebrates like reptiles, birds, mammals, and amphibians, and terrestrial arthropods. Human respiratory system. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is the common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps in sound production and hence called the sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity, which divides at the level of fifth thoracic vertebrae into a right and left primary bronchi. Each bronchi undergoes repeated divisions to form the secondary and tertiary bronchi and bronchioles, ending up in very thin terminal bronchioles. The trachea, primary, secondary, and tertiary bronchi, and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular walled and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles, and alveoli comprise the lungs. Two lungs, which are covered by a double-layered pleura with pleural fluid between them, are present in human body. It reduces friction on the lung surface. The outer pleural membrane is in close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The parts starting with the external nostrils up to the terminal bronchioles constitute the conducting part. That is, the pair of external nostrils, nasal passage, nasal chamber, pharynx, larynx, trachea, which extend up to mid-thoracic cavity and divides at the fifth thoracic vertebrae into left and right bronchi. Then primary bronchi, secondary bronchi, tertiary bronchi, pulmonary bronchiole, and finally terminal bronchiole. This together forms the conducting part or the bronchial tree, and the alveoli and their ducts form the respiratory or exchange part of the respiratory system. That is, the respiratory bronchiole, alveolar duct, and alveoli together form the respiratory tree or the respiratory or exchange part of the respiratory system. The conducting part transports the atmospheric air to alveoli, clears it from foreign particles, humidifies, and also brings the air to body temperature. Exchange part is the site of actual diffusion of oxygen and carbon dioxide between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by a vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by a dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung or pulmonary cavity. Such an arrangement is essential for breathing as we cannot directly alter the pulmonary volume. Respiration involves the following steps: breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2 rich alveolar air is released out. Diffusion of gases O2 and CO2 across alveolar membrane. Transport of gases by the blood, diffusion of O2 and CO2 between blood and tissues, utilization of O2 by the cells for catabolic reactions, and resultant release of CO2. Diagram showing human respiratory system. Epiglottis, larynx, trachea divide into bronchi or bronchus and the fifth thoracic vertebrae. Left lung, bronchiole, alveoli, double-layered pleural membranes filled with pleural fluid, cut end of rib, lungs, diaphragm from bottom, and heart in the mediastinum cavity. Mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, that is intrapulmonary pressure, is less than the atmospheric pressure. That is, there is a negative pressure in the lungs with respect to atmospheric pressure. Similarly, expiration takes place when intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal intercostals between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the antero-posterior axis. The contraction of external intercostal muscles lifts up the ribs and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs, that is inspiration. Now, relaxation of the diaphragm and the intercostal muscles returns the diaphragm and sternum to their normal positions and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs. This is called as expiration. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, healthy human breathes about 12 to 16 times per minute. A healthy human breathes for 12 to 16 times per minute. The volume of air involved in breathing movements can be estimated by using a spirometer, which helps in a clinical assessment of pulmonary functions. Diagram showing inspiration is given. Diaphragm contracts, ribs and sternum raised, volume of thorax increased, ribcage is shown, and air enters the lungs from external atmosphere. Here intrapulmonary pressure is negative with respect to atmosphere. Volume increase, pressure decrease, so air from atmosphere enters into the lungs. A diagram showing expiration. Ribs and sternum return to their original position. Diaphragm relaxed and arced upwards. Volume of thorax decreased. Air is expelled from lungs. Intrapulmonary pressure is positive with respect to atmosphere. Respiratory volumes and capacities. Tidal volume TV. Volume of air inspired or expired during a normal respiration. It is approximately 500 milliliter. That is, a healthy man can inspire or expire approximately 6,000 to 8,000 milliliter of air per minute. 6,000 to 8,000 milliliter of air per minute. Volume of air inspired or expired during a normal respiration is approximately 500 ml. Inspiratory reserve volume (IRV). Additional volume of air a person can inspire by a forcible inspiration. This averages about 2500 ml to 3000 ml. Expiratory reserve volume (ERV). Additional volume of air a person can expire by a forcible expiration. This averages about 1000 ml to 1100 ml. Residual volume (RV). Volume of air remaining in the lungs even after a forcible expiration. This averages 1100 ml to 1200 ml. Pulmonary capacity is used in clinical diagnosis. Inspiratory capacity (IC). Total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume. TV plus IRV. It's about 3000 to 3500 ml. Expiratory capacity (EC). Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume. TV plus ERV is equal to 1500 to 1600 ml. Functional residual capacity (FRC). Volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV, that is expiratory reserve volume plus residual volume. Vital capacity (VC). The maximum volume of air a person can breathe in after a forced expiration. This includes expiratory reserve volume, tidal volume, inspiratory reserve volume, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity. Total volume of air accommodated in the lung at the end of a forced inspiration. This includes residual volume, expiratory reserve volume, tidal volume, and inspiratory reserve volume, or vital capacity and residual volume. Vital capacity plus residual volume. Trick for respiratory volumes and capacities. Reti's friend Ileana is very tall. RETI. R stands for residual volume, expiratory reserve volume, tidal volume, inspiratory reserve volume. Residual volume is volume remaining in lungs even after a forced expiration. Expiratory reserve volume is the additional volume which can be expired by a forced expiration. Inspiratory reserve volume is the additional volume which can be inspired by a forced inspiration. Tidal volume is the volume inspired or expired by a normal respiration. Residual volume is about 1100 to 1200 ml. ERV is 1000 to 1100 ml. Tidal volume is 500 ml and inspiratory reserve volume is 2500 to 3000 ml. RV plus ERV forms the functional residual capacity, volume of air that will remain in lungs after a normal expiration. EC, expiratory capacity, is the volume of air a person can expire after a normal inspiration. It's tidal volume plus expiratory reserve volume. Inspiratory capacity is tidal volume plus inspiratory reserve volume. Volume of air a person can inspire after a normal expiration. Vital capacity is the maximum volume of air a person can breathe in after a forced expiration, or maximum volume of air a person can breathe out after a forced inspiration. It's expiratory reserve volume plus inspiratory reserve volume plus tidal volume. And total lung capacity is the total volume of air accommodated in lungs at the end of forced inspiration. Residual volume plus tidal volume, or RETI. Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. Oxygen and carbon dioxide are exchanged in these sites by simple diffusion, mainly based on pressure or concentration gradient. Solubility of gases as well as the thickness of membranes involved in diffusion are some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is represented as PO2 for oxygen and PCO2 for carbon dioxide. Concentration gradient for oxygen from alveoli to blood and blood to tissues. Similarly, a gradient is present for CO2 in opposite direction, that is from tissues to blood and blood to alveoli. Partial pressure in mmHg of oxygen and CO2 at different parts involved in diffusion in comparison to those in atmosphere. Concentration gradient for oxygen in atmosphere is 159 mmHg, for alveoli 104 mmHg, oxygenated blood 95 mmHg, deoxygenated blood 40 mmHg, for tissues 40 mmHg. For concentration gradient of CO2 in tissues is 45 mmHg, oxygenated blood 40 mmHg, deoxygenated blood 45, alveoli 40, atmospheric air 0.3 mmHg. As the solubility of CO2 is 20 to 25 times higher than that of oxygen, the amount of CO2 that can diffuse through diffusion membrane per unit difference in partial pressure is much higher compared to that of oxygen. The diffusion membrane is made up of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance composed of thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single layer endothelial cells of capillaries in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors in our body are favorable for diffusion of oxygen from alveoli to tissues and that of CO2 from tissues to alveoli. Diagram of a section of an alveolus with a pulmonary capillary. Alveolar cavity, blood capillary, alveolar wall, squamous epithelium of alveolar wall, it's one-celled thick, blood capillary, RBC, endothelium of alveolar capillary, basement substance, air, alveolar cavity, air. Basement substance composed of thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single layered endothelial cells of capillaries. Diagrammatic representation of exchange of gases at the alveolus and the body tissues with blood and transport of oxygen and carbon dioxide. Concentration gradient for O2 is alveolar blood tissue. Concentration gradient for CO2 from tissue blood alveoli. Air is inspired into the alveolus where partial pressure of atmospheric oxygen is 159 mmHg and in alveoli it's 104 mmHg. So oxygen moves into alveoli due to concentration gradient or pressure gradient. Partial pressure of oxygen in deoxygenated blood, which comes from pulmonary vein, is 40 mmHg. So oxygen moves from alveoli to the vein, passes into the heart and then through systemic arteries which carries oxygenated blood to the body tissues where partial pressure of oxygen is 40 mmHg. So oxygen moves from artery to the body tissue. Further, systemic veins carrying deoxygenated blood, partial pressure of oxygen is 40 mmHg. Now partial pressure of CO2 in body tissue is about 45 mmHg, whereas in oxygenated blood, which comes from systemic arteries, is about 40 mmHg. So CO2 from body tissues moves to oxygenated bloods and make it deoxygenated blood, where its partial pressure of CO2 is turns to 45 mmHg. This blood passes through systemic veins into the right auricle and ventricle and passes to pulmonary artery. This blood from pulmonary artery diffuses into the alveolus as partial pressure in alveolus of CO2 is 40 mmHg and is expired out in air where atmospheric pressure of CO2 is 0.3 mmHg. This cycle continues. Of gases. Blood is the medium of transport for oxygen and CO2. About 97% of oxygen is transported by RBCs in the blood. Remaining 3% of oxygen is carried in dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, whereas 70% of it is carried as bicarbonate and 7%, about 7% of CO2 is carried in a dissolved state through plasma. Transport of oxygen. Hemoglobin is a red colored iron containing pigment present in RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to partial pressure of oxygen. Partial pressure of CO2, hydrogen ion concentration, and temperature are other factors which can interfere with this binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with oxygen is plotted against PO2. This curve is called the oxygen dissociation curve. It shows right to left shift. Oxygen dissociation curve is highly useful in studying the effect of factors like PCO2, H plus concentration, etc. on binding of oxygen with hemoglobin. In the alveoli where there is high partial pressure of oxygen, low PCO2, lesser H plus concentration and lower temperature, the factors are all favorable for the formation of oxyhemoglobin, whereas in tissues where low PO2, high PCO2, high H plus concentration and higher temperature exist, conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that oxygen gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of oxygen to tissues under normal physiological conditions. Every 100 ml has 5 ml of oxygen. 100 ml oxygenated blood has 5 ml oxygen. Factors favorable for formation of oxyhemoglobin are high PO2 concentration, low PCO2, low H plus concentration and low temperature. Graph showing oxygen dissociation curve. Partial pressure of oxygen in mmHg is plotted on X scale and percentage saturation of hemoglobin with oxygen on Y scale. At partial pressure of oxygen 100 mmHg, about 100% saturation of hemoglobin with oxygen is seen. Transport of carbon dioxide. CO2 is carried by hemoglobin as carbaminohemoglobin, about 20 to 25%. This binding is related to partial pressure of CO2. PO2 is a major factor which could affect this binding. When PCO2 is high and PO2 is low, as in the tissues, more binding of carbon dioxide occurs, whereas when the PCO2 is low and PO2 is high, as in the alveoli, dissociation of CO2 from carbaminohemoglobin takes place. That is, CO2 which is bound to hemoglobin from the tissues is delivered at alveoli. RBCs contain a very high concentration of enzymes, carbonic anhydrase, and minute quantities of same is present in the plasma too. This enzyme facilitates the following reaction in both directions. CO2 plus water in presence of carbonic anhydrase gives carbonic acid H2CO3, which further converts to bicarbonates and H plus ion in presence of carbonic anhydrase. This is a reversible reaction, which means bicarbonates and H plus can further be converted to H2CO3 carbonic acid in presence of carbonic anhydrase, which can further be converted into CO2 and water in presence of carbonic anhydrous. At the tissue site where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, RBC and plasma and forms bicarbonates and H+. At the alveolar site where PCO2 is lower, the reaction proceeds in the opposite direction, leading to formation of CO2 and H2O. Thus, CO2 trapped as bicarbonate at the tissue level and transported to alveoli is released out as CO2. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. 100 ml deoxygenated blood has 4 ml CO2. Chloride shift takes place where bicarbonate ion is replaced by chlorine ion. HCO3 minus is replaced by Cl minus and H plus binds with hemoglobin and forms HHB. Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by neural system, a specialized center present in the medulla region of the brain called respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the functions of the respiratory rhythm center. Neural signal from pneumotaxic center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to rhythm center which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate chemosensitive center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process by which these substances can be eliminated. Receptors associated with aortic arc and carotid artery also can recognize changes in CO2 and H plus concentration and send necessary signals to the rhythm center for remedial actions. The role of oxygen in regulation of respiratory rhythm is quite insignificant. Role of oxygen in regulation of respiratory rhythm is quite insignificant. The respiratory rhythm center in medulla region is primarily responsible for regulation of respiratory rhythm. This is a specialized center. Pneumotaxic center in pons region can moderate functions of respiratory rhythm center. Neural signal from pneumotaxic center can reduce duration of inspiration and thereby alter respiratory rate. Adjacent to rhythm center is a chemosensitive area which is sensitive to increase in CO2 and H plus ions. Increase in CO2 and H plus activates this center and it signals the rhythm center to make adjustments in respiratory process to eliminate these substances. Now, receptors associated with aortic arc and carotid artery recognizes changes in CO2 and H plus concentration and send necessary signals to rhythm center for remedial actions. In all the three pneumotaxic center in pons region, chemosensitive area adjacent to rhythm center, and receptors associated with aortic arc and carotid artery overall affects the respiratory rhythm center in medulla region, which is specialized center for regulation of respiratory rhythm. CO2 and H plus concentration is significant in regulation of respiration. Disorders of respiratory system. Asthma is a difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged due to which respiratory surface is decreased. One of the major causes of this is cigarette smoking. It causes rupture of alveoli. Occupational respiratory disorders. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of body cannot fully cope with the situation. Long exposure can give rise to inflammation leading to fibrosis, proliferation of fibrous tissues, and thus causing serious lung damage. Workers in such industries should wear protective masks. Occupational respiratory disorders, fibrosis, proliferation of fibrous tissue. dyspnea, difficulty in breathing, atelectasis, collapsing of lungs, pneumonia, inflammation of air sacs due to bacterial or viral infection, S. pneumoniae. Summary: cells utilize oxygen for metabolism and produce energy, along with substances like CO2, which is harmful. Animals have evolved different mechanisms for the transport of oxygen to the cells and for removal of CO2 from there. We have a well developed respiratory system comprising two lungs and associated air passages to perform this function. The first step in respiration is breathing, by which atmospheric air is taken in, inspiration, and the alveolar air is released out, expiration. Exchange of oxygen and CO2 between deoxygenated blood and alveoli, transport of these gases throughout the body by blood, exchange of O2 and CO2 between the oxygenated blood and tissues, and utilization of oxygen for the by the cells. Cellular respiration are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscles, intercostals and diaphragm. Volumes of air involved in these activities can be estimated with the help of spirometer and are of clinical significance. Exchange of O2 and CO2 at the alveoli and tissues occurred by diffusion. Rate of diffusion is dependent on partial pressure gradients of O2, PO2 and CO2, PCO2, their solubility as well as the thickness of diffusion surface. These factors in our body facilitate diffusion of oxygen from alveoli to deoxygenated blood as well as from deoxygenated blood to the tissues. The factors are favorable for the diffusion of CO2 in the opposite direction, that is from tissues to alveoli. Oxygen is transported mainly as oxyhemoglobin. In alveoli where PO2 is higher, O2 gets bound to hemoglobin, which is easily dissociated at the tissues where PO2 is low and PCO2 and H plus concentration are high. Nearly 70% of carbon dioxide is transported as bicarbonate HCO3 minus with the help of enzyme carbonic anhydrase. 20 to 25% of carbon dioxide is carried by hemoglobin as carbaminohemoglobin. In the tissues where PCO2 is high, it gets bound to blood, whereas in the alveoli where PCO2 is low and PO2 is high, it gets removed from the blood. Respiratory rhythm is maintained by the respiratory center in the medulla region of brain, a pneumotaxic center in the pons region of the brain, and a chemosensitive area in the medulla can alter respiratory mechanism.